So we've touched on some of these topics before, but now I want to discuss them sort of in detail. The question of what impacts results that you get from your model. And there's certain types of uncertainty, and they can, more than one can come into play that can affect the results from your models. You'll see that when you read papers, most of the time these uncertainties are not actually explicitly acknowledged, but people in the field understand that. Like, to some extent, every scientist understands that when they study a certain system, there are limitations which might or might not translate to the real world in some sense. That's true at every level. Even if we do things like clinical trials for humans, it's hopefully a very good approximation of the real system, which is the general population, but it's still, in some sense, abstract. And of course, the more you go to the bench sciences, and then to modeling, the more abstract the system becomes. So an important component to look at is structural uncertainty. And by that, what I mean is that you build your model and it has a certain structure. And we've mentioned that already multiple times and discussed it, that it's always a simplification of the real world. And you have to decide which components to include and which components to exclude and what assumptions to make. So there's sometimes this flippant saying every model is wrong, which is true. You can, in some sense, it's always an approximation of the real world. Wrong doesn't mean in the sense that it's useless. It just means it's not exactly the real system. And we actually want that. We want this approximation, because if we had a model that was exactly the real system, it would be as complex as the real system, and then we wouldn't have gained anything. So we need to make approximations, and we need to decide what do we include and what to not exclude. I like thinking of it in terms of maps, and so this is a figure that nature drew for us in our paper there. This is a made-up country, of course, that they called Antipodia. The point here is that depending on what question you have, even though it's the same map, or it's the same country, one map might be better than another. So if you know where this country is in relation to other countries, the map on the left would be best. If you want to know how to go from place A to place B in that country, you probably want the middle map. If you want to know where people live in that country, the one on the right one is the best. So same map, if we translate that, same model, same system, but depending on what your question is, you might prefer one over the other. And so it's the same when you build your models. You have to decide which parts do you include, which parts do you exclude. And there's no rule that says, okay, if we model a virus, we need to include that. If we model malaria, this needs to be in there. It's true that there are certain things that are more likely to be included or not, but it's really mostly driven by the question. So you could study the same system, different questions, and you might come up with very different models. Someone isn't muted. Could everyone make sure they're muted, please? I can't unfortunately see it, otherwise I could mute everyone. Okay. So let's talk about types. Let's talk about structural uncertainty a little bit more, and the types of structural uncertainty you can have. So one thing is you decide which components to include. Do you want to include T cells? Do you want to include both CD8 and CD4 T cells? Do you need the innate response? Do you need every cytokine individually or only important ones? So these are the components you want to include. And we've already discussed that a little bit. And then the mechanisms and processes. We looked at a very simple one initially where we said, well, we could model exponential growth or we could model linear growth. And basically the same considerations you can apply to every term in your equation. You can say, okay, do we assume mass action where they bump into each other, or do we need to include some kind of saturation in there? So these are decisions you need to make. And then another one that we really haven't covered, however, there's a reading on the webpage that I hope you've been able to go through what type of model. Here we're looking at these compartmental models, mostly formulated as differential equations, but there are different types out there. You have these agent-based models. You could have network models. You can have all kinds of models. And so what type of model to use? And all of these decisions can potentially affect the results. Unfortunately, there's no a priori way to know what affects the results and by how much. So to some extent, it's a bit of a trial and error. The problem is you can't systematically test it. When you build your model, you might be able to explore a few variants. You could include or exclude certain components. You could formulate a few processes in different ways, but you can't exhaustively cover everything. That would just be way too much. So just mention that. Here's an example where we have growth either exponential or linear. And here we have one where we have either mass action or some kind of saturated killing, where basically when B is really large, this term approaches 1. So there's a maximum rate of killing by the immune response, no matter how many bacteria there are. So these are all choices one has to make when one builds models. The considerations I think that are useful is biology. So if you know that a certain process is exact or very likely a certain way, you might want to model it that way. Or if a certain component is really important, you should include it. And then often simplicity and parsimony. So you go with the simple model, the one that has the fewest parameters that you think captures the processes in the system decently enough. Here's an example from a paper that I thought was a really nice one. What they did here, they did not themselves build models, but they pulled other people's models, and these were all models for influenza. And those models were developed for different purposes. They included and excluded different things. None of these models were the same. And they actually compared them to data. So in the top, you see different models, and you see that not every model is in every plot. That means that's the reason, because some models didn't actually include that component. So you can see there's this black one called Bokharov. They named it by the first author, and that is included in each one of these plots because it has all these components in there. But there's, for instance, that green model called BACAM that's only in the right panel because it doesn't have antibodies and T cells included in the model. So different groups made different choices in which components to include and which components to exclude. And then they compared the models that did have that component in there to data. So at the bottom, you see data collected from different experiments. They went through the literature, they looked at it, and tried to see, okay, which models can capture which patterns. And it's worth going through the paper, but basically the upshot is no single model was good at explaining all the data. So they were all able to do certain things and not so good at certain other ones. But that comparison itself was a whole paper and probably took a good bit of work. Now, if any individual person, when they were developing a model, had tried all these combinations, it would have just been a whole lot of work and maybe not so useful because they wanted a model to address a certain question. And so including components that weren't useful for their question might not be useful. So, it's important to keep in mind that as you make your model choices, it might affect your outcomes. And it's really the biggest source of variability in outcomes. It's kind of unfortunate that this is the biggest source and it's the least studied because there is no systematic way to deal with it. You can compare across models. This is very rare on the within host level. It's more common on the between host level now. And a good topical example is actually this link I put in there, which is the CDC's webpage, where there's a bunch of population level coronavirus models are being used to forecast, and then they're showing them all on that site. So they're not actually comparing the insides of the model, but they're comparing the model predictions and actually building an ensemble model that takes the average of those models. There have been other efforts like that on the population level for HIV and TB, where multiple independent modeling groups all studied the topic and all were asked to, predict the same outcomes and results, and then the models were combined, or the results were combined to look at some changes across models. And what you gain from that is that if there's a certain result that is consistent across all models, then it's pretty solid evidence that this might be a real effect. If there's something else where every model says something different, you probably can't put too much trust into that. And again, to make the point that this is not just specific to these kind of simulation models, it's exactly the same in any kind of experimental model. You know, if you do an in vitro experiment and you take 5 different cell lines and every cell line gives you a different answer, you should probably not take any one of those cell lines and take those results and say this applies to humans. Now, if all 5 cell lines give you the same result, you could say, well, this is maybe a more robust result. Or you have 5 different mouse strains that you use. Now, again, no one really does that because this is time consuming and expensive, and you might do it in 1 or 2 or maybe 3 systems, but then you stop at that point. That's just an inherent, feature we have to live with, both for computational models like the ones we discuss here and for any other kind of model system that we have not we have no way of efficiently systematically exploring across all those models. So that's important to keep in mind when you both build your models, but also when you read results from models in the literature. The results they're presenting are contingent on the choices they make when they build their models, and you can read it and can decide that you thought they made reasonable choices and therefore you might be able to trust their model, or you decide that they excluded important components or didn't do the model formulation right and therefore you're not quite trusting what they're presenting. But this is always a feature. There's just nothing we can do about it other than fully acknowledge and fully explain that that comes with any model.